Good morning, students. Hope you are doing good at home, enjoying the holiday slash quarantine slash, stay at home stage. Uh, the main purpose is to be safe uh, for ourselves as well as for others. So the stay at home option is actually good for all of us. So I know that some of you might be thinking that we need to go out, but just control it for some more days. I think the situation will be under control and we all can go out and roam around independently. So, anyway, sorry about posting this recording a little later, but this is going to be really interesting lecture, definitely. So let's start from here where we stopped. So we stopped at RAN independent nuclear export, and before that, we saw RAN dependent nuclear export, where we had RAN gap and RAN Jeff concentration, and depending on that, there was unidirectional transport of proteins across the nuclear membrane, or rather NPC, nuclear pore complex. Can you tell me where exactly was more concentration of RAN gap, cytosol or nucleoplasm? If you can tell me the answer, then you're good. If you cannot, we need to go ahead and read that or listen to that lecture again. By the way, this presentation is pretty much same as the last presentation, but it is newly uploaded with where we stopped as the marking between the third and the fourth lecture. So you can check it out. Anyway, so this particular slide we discussed about RAN independent. So there is no RAN involvement is there, but there is NXF and NXT1, which acts as nuclear receptor, transport receptor instead of exportin 1. And these factors help only the processed mRNA. So that means this process is only for processed mRNA and nothing else. So processed mRNA gets bound by these receptors, and then they take this mRNA out of this NPC and they get recycled back with the help of energy produced by the ATP hydrolysis by DBP5, which is a helicase and is present near the NPC outside, like towards the cytosolic surface. Okay, let's go to the next lecture, lecture 4. So lecture 4 talks about vesicular traffic, secretion, and endocytosis. So far, what we have seen is that the proteins which are unfolded gets inside either the ER or folded version proteins gets into the mitochondria or chloroplast or nucleus, depending upon what kind of signals or targeting signals they have. In this particular case, we will be talking about the vesicular trafficking and the secretory proteins. Secretory proteins are the proteins which basically gets inside the endoplasmic reticulum or ER and then gets modified or folded there and then goes to the next compartment, which is the Golgi or rather Golgi network, and then from there it can have two options: either go and get secreted either on the plasma membrane or outside, or it can go to the lysosome. So today we'll be discussing about how the proteins gets transported in this through this secretory pathway. Okay, so there are different techniques that have been used to study these secretory pathways and to get the information exactly what's happening. Okay, so the first technique use the various labels enable tracking protein movement through the secretory pathway to different destinations. As I mentioned, different destinations can be either to the next set of network, that is Golgi network, or the plasma membrane, or it can also be the lysosome. Okay. So various labels, mainly labels means it can be dye or fluorescent tags, etc., have been used. We have at UTD Biological Sciences Department a lab. This lab is actually Dr. Lee Zhang's lab, who joined UTD in 2008. Her lab actually studies about these protein movements by using tracking dye. So basically what her lab does is they create hypoxic environment for yeast cells. So hypoxic environment means less oxygen than the required amount. So when there are hypoxic environment in the cell, what cells does, they secrete certain specific proteins. Also, certain proteins gets transported and gets translocated into certain regions or organelles rather than their usual. So what she does, she compares between the normal proteins in normal case scenario and where they are going, and in case of hypoxic environment, what kind of proteins move to what kind of compartments. And she does that by tracking those proteins by using certain drugs. I mean, not drugs, sorry, certain dyes. And we can actually visualize those particular proteins tagged by those particular dyes when they move from one compartment to another compartment after some time. Okay, so there is one. The components required for intracellular protein trafficking have been identified by analysis of yeast temperature sensitive secretory mutants. It's a long name. Short name is SEC mutants, SEC, SEC mutants. Okay. This is also interesting because we are going to see how interesting is this based on we can change the temperature and the mutant protein will stop at certain compartment and do not move to the next one, which is unique. So we can easily study those particular proteins getting stuck at certain compartments. The third is the cell-free assays for intercompartmental protein transport. So cell-free assays also have been used. So these three techniques have been used. So let's go to the general process. This is the general process we were talking about. So this is overview of secretory and endocytic pathway of protein sorting. Okay. So secretory pathway involves ER and then this network of Golgi. I say network because this is cis Golgi and then medial Golgi and then trans Golgi and then trans Golgi network or short form TGN. Then from here, it can either go to the cytosol, I mean, not the cytosol, but towards the plasma membrane, or it can go and get secreted outside the plasma membrane, that means outside the cell itself, or sometime it can also go to the lysosome. Okay, so let's talk about the first step, what's happening. The stage one is basically the production of protein, not production, that's a wrong sentence. The folding of proteins inside the ER lumen. We know that the production of proteins or the synthesis of proteins, it's a co-translational translocation process for ER. So as the peptide is getting produced or synthesized with the help of ribosome on the surface of ER, they are also getting inside the ER. Once they get inside the ER, the signal sequence gets cleaved inside the ER lumen, and then the protein can get folded into its proper structure, functional structure. Okay. Now, from there, the next steps, that means all these steps are involved, that is called protein trafficking. Okay, so the first is the synthesis and folding of peptides here inside the ER lumen, and then the second stage is all these steps, that is protein trafficking. So in case of protein trafficking, what happens is that these ER lumen, these not ER lumen but ER walls, can actually bud off. Bud off means they form smaller vesicles, and those smaller vesicles basically resembles the ER itself, a smaller version of ER itself. Can you remember what other structures resembled ER? We studied them before. Think about it. Got it? It is microsomes. So basically, when we blended these ERs, we basically got microsomes. Those were smaller circular versions of ER. And based on Boolean density, we could have isolated them from other organelles. Okay. Anyway, so here, this is the natural process inside a cell where a vesicle forms, not a vesicle forms, but vesicle keeps on forming and keeps on budding off. And as they form, they also take these folded proteins with them. And they eventually form this cis Golgi, rather a small composition of buds forming a bigger structure, which eventually forms cis Golgi. So since I said it keeps on budding vesicles and keeps on forming this structure, that means this is one layer, this goes here, this becomes the second layer, and then other layers we'll discuss. That means this kind of layers are keeps on forming from this ER. Okay. Now what happens? Well, the second step is the budding and fusion of ER to Golgi vesicles to form cis Golgi, which is this step. The third step we are not going to talk about right now, but rather we are going to talk about the fourth step. Fourth step is cisternal maturation. Now this is interesting because we can see these smaller vesicles getting budding off from the ER and then forming this kind of cis Golgi structure. But then there are no vesicles budding off from cis to medial or medial to trans. So here what happens is that there are certain enzymes which are specific to these components. Let's say these existed in the beginning. These cis Golgi, medial Golgi, and trans Golgi, they existed in the beginning of the creation. Let's assume that. Okay. Now what happens is that trans Golgi have certain specific enzymes which basically makes the whole set as trans Golgi. Medial Golgi has certain specific enzymes which gives the characteristic feature of medial Golgi, and so does the cis Golgi. So what happens, trans Golgi rather buds of vesicles along with proteins and the specific enzymes for trans Golgi, and they basically come back and fuse with this medial Golgi. And so medial Golgi now have the enzymes mainly from the trans Golgi. So ideally it should become the trans Golgi, but not yet because there is still some medial Golgi present. Okay. I mean, some enzymes for medial Golgi present in the medial Golgi. So in the medial Golgi right now, we have the enzyme specific to trans Golgi as well as enzymes specific to medial Golgi. Now what happens at the same time when these enzymes are basically getting added to these bud of vesicles and getting fused here. The same time, the medial Golgi also bud of vesicles containing the enzyme specific to the medial Golgi, and they fuse back with the cis Golgi. So that means when this is moving its enzyme here, this is moving its specific enzymes here. So at some point, this will only have the enzyme specific to trans Golgi. And as I discussed before, the enzyme specific enzymes gives the characteristic feature of the compartment. So basically this medial Golgi becomes trans Golgi. This is unique, right? So at the same time, when this medial Golgi is moving its enzyme to cis Golgi, at the same time, the cis Golgi is also moving its specific enzymes to this cis Golgi network. Okay, so that means that cis Golgi now becomes medial Golgi. Okay, and then as I mentioned that these vesicles continuously gets bud off to form this kind of structure. So by the time all of these are happening, there is another layer or another vesicle that forms here that becomes the cis Golgi network with the actual cis Golgi specific enzymes. So that is how the cisternal maturation happens through backtracking of the specific enzymes, compartment specific enzymes. Okay, this is cisternal maturation. We'll also discuss this later on, but this is how the cisternal maturation happens. Now, this is the fifth step. What was the third step? Well, third step I did not tell you before because this is the backtracking or retrograde transport. So when we are moving up, that is the natural order, that is the ER lumen to cis Golgi network to cis Golgi, medial Golgi, trans and trans Golgi network. This is called anterograde transport. But when we are moving back, these are called retrograde transport. Okay, so sometime what happens is that, let's say I mentioned before that folded proteins gets inside these budding off vesicles and gets fused to cis Golgi network. But sometimes, there can be possible that we can have some misfolded proteins which also gets into these excuse me, gets into these vesicles. What happens then? They have to be returned back to the ER because they need to be fully folded and properly folded. Only then they can go forward. Okay, so basically it's like training process, multi-step training process. If let's say there are like 4 stages of training, A, B, C, D, and you couldn't pass training level A, you cannot go to training level B, okay, because you won't be successful. So if someone fails at training level B, that person has to repeat the training level A, okay, just like that. So the proteins basically backtracked or through the retrograde transport comes back from Golgi ER. Okay, now comes this fourth, and then now comes this fifth step. We are done with that. Through this anterograde transport, what happens? It goes to the trans-Golgi network. This trans-Golgi network is unique, and we will see why it is unique, because it has the capability to form coat proteins. We are not going into coat proteins right now, but just know that it is unique. Okay, now from here it has two options. Okay, it can either go through this, what is the six? Yes, it can either go to the constitutive secretion process, or it can go through this regulated secretion process. What is constitutive secretion? Constitutive secretion means continuously these particular uh, vesicles are budding off from this TGN, and they are going into this plasma membrane, and it's happening continuously. Okay, there is no stoppage. Which also, these vesicles also contain secretory proteins. Remember, the vesicles are not that important, but what it carries is more important. So it carries these secretory proteins that we require for the regular cellular functioning. Okay, so these proteins can either go and becomes the plasma membrane proteins. So in that case, these vesicles will fuse with these plasma membranes, and these proteins will become part of the plasma membrane itself. Remember, there needs to be certain channel proteins and all on the surface, receptor proteins on the surface. So basically, these secretory proteins becomes those membrane proteins. And some proteins actually gets transported outside the cell, that is in the intercellular spaces. Remember the endocrine hormones? Just a second. Yeah. So remember the hormones, those basically get secreted to the bloodstream. So those, the peptide hormones and all, those can be the secreted proteins. Okay. Now, what about this seventh step, that is regulated secretion? This is unique. I mean, everything is unique to me. So this is unique in the sense that the whole process is regulated. Now you can say that why? The reason is the cell doesn't expend energy unless it is required. It is extremely intelligent system. Okay, everything has to be to the point. So certain proteins need to be secreted at larger quantity when the time comes. Unless the time comes, there is no need to have those proteins. So although these proteins gets into these vesicles, but they don't get secreted out. Exactly when the need comes, certain kind of signals, extracellular signals, are received, and then only these secretory vesicles will go and fuse with the plasma membrane and release their proteins outside. That is why it is called regulated secretion. Now, the eighth step, there is the vesicles which do not go through either constitutive secretion or the regulated secretion. They go to these lysosomes. Why? Well, we said that proteins gets properly folded here. True. Sometimes, I mean not sometimes, the proteins have to have certain oligosaccharide side chains as well, which gets added by these membrane specific, not the membrane, but the compartment specific enzymes, and those determines how the protein gets matured because of the presence of specific compartment specific enzymes, right? So sometimes it may happen that some of the proteins do not get properly uh, processed. We won't say folded, but processed. So if sometimes some of the proteins may not get properly processed, but might somehow skip into this TGN. But the cell knows that we cannot release an untrained soldier outside. So what it does, it basically sends the soldiers back. That is one process. The other is we basically discard the soldier. In this case, we have to destroy those proteins. So what happens? These basically vesicles bud off. They mix with this structure or the organelle called late endosome. Late endosome is specified by the presence of low pH, about 5. Okay, pH 5. That means it has an acidic nature. So proteins go here, vesicles gets fused with late endosome, and then late endosome itself fuses with a lysosome. And we know that lysosome also has acidic environment, as well as lysosome has certain proteases which completely denatures or destruct the protein. Okay, so that is one pathway. There is this sorting pathway through the step 8 lysosome destined membrane and soluble protein pathway. The last one, ninth one, This is called endocytic pathway. Endocytic pathway, here the proteins are not involved which originated inside. Rather, those are the extracellular proteins. Now remember, I said that sometimes through these regulated secretion, when the time comes, specific situation arrives, there are extracellular signals, and when the cell receive those extracellular signals, these regulated regulatory secretory proteins basically get secreted outside. When the function is done, that means the situation has been overcome, then what will happen to those proteins, because those proteins are not needed anymore. Again, as I mentioned, cell cannot expend more energy on anything. Okay, it is very precise system. So those proteins, as well as, let's say, certain proteins whose function has been already done, the protein has been expressed, and sometimes there are more proteins. So though all those proteins, or sometimes there are somehow there are misfolded proteins, somehow escaped. So all those proteins needs to be taken inside to get destroyed. So basically, the inside invagination, instead of budding off, the plasma membrane invaginates to form these vesicles. These vesicles are called endocytic vesicles, which eventually goes and makes these late endosome by fusion. Then late endosome basically fuses with the lysosome. The whole purpose is to bring back those proteins which are not required anymore, to get them destroyed inside the lysosome. Okay, so if I ask you what is the difference between the constitutive secretion and regulated secretion, what would be the answer? The answer is constitutive secretion continuously secretes secretory proteins outside or on the plasma membrane. Regulated secretion regulates the secretion mechanism of secretory proteins, and when there is a need, only then through this extracellular receptor or through these extracellular signaling, the regulated secretion happens. Okay, so that is the whole general overview of secretory and endocytic pathway of protein sorting. Let's now talk about specifically the secretory pathway of protein sorting. And for that, remember we use three techniques, or we mentioned about three techniques. So let's see this first technique. This first technique is protein transport through the secretory pathway can be visualized by fluorescence microscopy of cells producing a GFP tagged membrane protein. Okay, this GFP tagged membrane protein that is being used here is called vesicular stomatitis virus G protein, or VSV G protein. It's a typical type 3 viral membrane fusion protein. We won't go into details about VSV-G protein because that's a whole new chapter itself. Anyway, so what happens? We are talking about fluorescence microscopy. So in this experiment, since this is the plasma membrane protein, the VSV-G protein, so we label the VSV-G protein, we basically fuse with the GFP, that is VSV-G GFP chimeric protein. Chimeric proteins means two proteins getting combined together, okay, and expresses as a whole. But we have to make sure that this tag, this GFP tag, also known as green fluorescence protein tag, does not affect its function, okay. So depends. Well, it depends, because if we are just looking at the location, then it doesn't really matter whether the tag itself affects the functionality of the protein. But if it is a functional study, then we have to make sure that any kind of tag should not affect the functionality of the protein, in this case VSVG. So GFP is green fluorescent protein which was discovered by a scientist through a jellyfish. So the scientist actually went on to a vacation and went on to do scuba diving and saw these jellyfish which were glowing inside the ocean. And he was like, wow, what is very specific in this? So basically, he took the jellyfish and then he extracted the protein, isolated that, and basically characterized the protein and used it in different cellular experiments, basically. And then a lot of cellular functioning were discovered using this particular technique, GFP tag. That particular scientist also received a Nobel Prize for his innovation. So it's not that when we go to a vacation or are relaxed, we are not thinking. If you have a scientific bent of mind, you will keep on thinking about things and you will keep on asking questions. Why is this happening? Why is that happening? Sometimes we get the answer, sometimes we don't. When we don't get the answer, we try to get the answer, and that is all about learning. Okay, anyway. So this protein was used in this particular case. And since this is a membrane protein, this VSVG protein, in this case, what we know that membrane proteins are form of secretory proteins. That means they have to be produced in the ER or folded into the ER, and then they have to go through this secretory pathway, that is ER to Golgi and then Golgi to plasma, right? We are skipping the network of Golgi here, but overall ER to Golgi, Golgi to plasma. Now, there is a unique situation here that in the ER, the restrictive temperature is at 40 degrees centigrade. That means if we increase the surrounding temperature to 40 degrees centigrade, in that case, the proteins cannot move out of the ER to the Golgi. That means none of the secretory proteins will go to their uh, required places. It also means, if you remember, if you are working in a microbiology lab, we sometimes, to inactivate the cells, we sometimes basically increase the temperature. That means it's a restrictive temperature. Even the temperature difference between 2 to 3 degrees centigrade can make a huge difference. This is just one example to show how restrictive temperature works, because all these secretory proteins, and there are like thousands of proteins, secretory proteins, which goes to have their own specific functions. If they do not get released to their specific areas or specific compartments and becomes functional, then the cellular functioning will not work anymore. It will either, the cell will either die or remain inactive. Okay. So anyway, this restrictive temperature is 40 degree centigrade. But when we change the temperature to 32 degrees centigrade, then it functions normally. Okay. So what was done is that we basically got the proteins, secretory proteins that we specifically know are supposed to go to the plasma membrane. We increase the temperature. We tag the protein, we increase the temperature. When we tag the protein is still inside the ER, and we can actually see it through the fluorescence microscopy that it's still in the ER. As soon as we change the temperature to 32 degrees centigrade, which is a permissive temperature, the protein starts moving outside and goes to the Golgi. That shows after 40 minutes. That shows that protein. So since these were GFP tags, we could actually track the movement of the proteins from one compartment to another. So it moved to this cis Golgi, or rather the Golgi, and then after 180 minutes altogether, it moved to plasma membrane. Okay, so from this experiment, tracking experiment, we can definitely say that the protein, the secretory protein, actually moves from one compartment to another with time. If we just graph this particular experiment here, we can see that this ER, this is the restrictive temperature at 0 minute, 42 degrees. So all the proteins are in the ER. As soon as we take the cell into permissive temperature, so from this point onwards till forward. Throughout this is the permissive temperature. So as soon as we change the temperature, immediately all the proteins basically gets out of the ER. That means the concentration of protein in the ER decreases immediately. As the concentration is decreasing, what is the next step? The next step is Golgi. As the concentration in the ER is decreasing, the concentration is in the Golgi is increasing at the same time because all the proteins are getting transported to the Golgi. And then as it increases in number in the Golgi, at the same time, proteins are also being transported to the plasma membrane. So once all the proteins are here, so all the proteins slowly starts moving to the plasma membrane, and by the time the concentration in the Golgi decreases. And when it goes to the plasma membrane, then either they get secreted or they stay on the plasma membrane. Now we are seeing that there is this decline in this graph, which is shown by the blue line. It could have stayed something like straight, or it could have stayed something like this. Well, not really, because protein concentration decreases from plasma membrane because some proteins gets, well, in this case, it should not, because it is a plasma membrane protein. So there is no question of secreting outside. So why is it decreasing? Think about it. Well, it decreases because we are looking at the GFP fluorescence through under the microscopy, fluorescent microscope. So the fluorescence actually reduces over time. That is why, although all the proteins are now at the plasma membrane level, the fluorescence slowly decreases. So we see the decline in the fluorescence. That is why this line is declining. Okay. That is all about this protein movement from one compartment to another with the help of fluorescence microscopy. Next, go to this slide, which basically mentions what happens and what will happen if the normal situation is disturbed. So we have a protein here. The protein is basically this peptide. What is this associated structure? This associated structure is oligosaccharide side chain. Okay, this oligosaccharide side chain is made up of N-acetyl glucosamine, also known as NAG, and then mannose residues, these blue balls here. Okay, blue structures here. So this is at 40 degrees centigrade. This is at 32 degrees centigrade. If we keep the temperature at the restrictive temperature at 40 degree, all the proteins will stay or remain staying in the ER. If we change the temperature to permissive temperature, now the proteins can move to the cis Golgi. During this movement process, what happens in the cis Golgi, there are certain, excuse me, excuse me. In the cis Golgi, mannose streaming happens. Okay, mannose streaming happens because mannose means these blue structures. You can see that is called maturation. Okay, so we can see how many units are there: 1, 2, 3, 4, 5, 6, 7, 8 mannose residues. Here we have 1, 2, 3, 4, 5, only 5 mannose residues, right? Now, what scientists did is that they basically extracted the glycoprotein along with the fat, obviously attached. So here is mannose 8 subunits, and glucnag 2 subunits, these red boxes. And here, excuse me, in this case, we have mannose 5 because mannose streaming happened. And then we have 2 glucnag residues as it is. Now, both of these were treated with endoglycosidase D. Okay. Now, endoglycosidase D, what it does is basically it's an enzyme that releases the oligosaccharide from glycoproteins or glycolipids. Okay, in this case, it is supposed to release this oligosaccharide from this glycoprotein, right? It may cleave towards the end, but usually it cleaves at the junction of oligosaccharide and the peptide itself, mostly or more commonly. Okay. So what happens is that protein moves from ER to cis Golgi, gets streamed, and then endoglycosidase D cleaves the oligosaccharide side chains from proteins processed in the cis Golgi. So only when the proteins are processed at the cis Golgi, only then this endoglycosidase D can be effective to remove this oligosaccharide side chains from the VSVG protein in this case, because this is secretory protein. But if this protein is not processed in the cis Golgi, this endoglycosidase D won't be effective, won't be able to cleave it here. So if the ER, if we isolate this whole assembly from the ER at restrictive temperature, since this particular assembly did not get processed, since it did not go to the cis Golgi to be able to get processed, so this will not get cleaved at this junction. So if we run these two kinds of peptides in the SDS page, what are we going to see? Well, this is a autoradiograph of the SDS page or polyacrylamide gel electrophoresis. So this is an X-ray film or nuclear emission film. So basically what we are looking at is this is the permissive temperature at different time points. So these are ER resistant proteins. These are uncleaved, larger and slower migrating proteins. But as the time increases, more and more proteins in the permissive temperature zone will move towards the cis Golgi. And as move to the cis Golgi, it will get processed. That means 3 mannose residues will get removed, and we can see a lighter or cleaved or smaller version of faster migrating protein. So that kind of protein concentration will increase as the time increases. This one is the electrophoresis data. Excuse me. It shows the percentage of endo-G sensitive VSV-G protein over time. So this at the restrictive temperature, all the proteins or most of the proteins stay in the ER. But as we go take it to the permissive temperature, most of the proteins get digested by endoglycosidase D, right? As time increases, more and more and more, eventually all of the proteins which went to the cis Golgi and got processed will get cleaved. So this particular experiment shows that membrane glycoproteins from the ER to the Golgi can be assayed based on sensitivity to cleavage by endoglycosidase D. Okay, only when the membrane glycoprotein is processed at the cis Golgi. Okay, now go to the next slide, and we were talking about these sec mutants. Now this is very, very interesting. Okay, cell-free assay demonstrates protein transport from one Golgi cisterna to another. So far we discussed about general protein movement from ER to Golgi to plasma membrane, and then we discussed about ER to cis Golgi. Okay, and then we are talking about how it goes and what is the other technique that we can use to know where the proteins are moving. Okay, so before that we need to know two terminologies. One is called NAG, There is N-acetyl glucosamine, and the other one is NAN. But before that, here, this is about temperature sensitive sec mutants. So 2 points to consider. One is called temperature sensitive. That means either it is a permissive temperature or a restrictive temperature. The other one is sec mutants. Okay, these are yeast sec mutants basically. Excuse me. Excuse me. So there are 5 stages in the secretory pathway that was discovered by these sec mutants. So what happens is that if it is a normal protein, normal secretory protein in the yeast, we can see the proteins all over the place with time. Obviously, some proteins gets produced in the ER or folded and then move to the Golgi, move to the vesicles, go out, and either becomes to the plasma membrane or go out. That is the normal secretion process. If it is mutated, that is class A mutant, in that case, they are accumulated in the cytosol itself. Otherwise, in normal case scenario, they could have been transported into the ER, but if it is a sec-A mutant and we increase the temperature to a restrictive temperature, that is why we use this term temperature sensitive. Then this particular kind of mutants, here the secretory proteins accumulate in the cytosol rather than going into the ER. Okay. The class B mutants, sec mutants, in this case, the proteins remain accumulated in the rough ER. Otherwise, in normal case scenario, they would have gone to the budding vesicles from the rough ER to be transported to the cis Golgi. But in case of sec B mutant at restrictive temperature, they stay in this rough ER itself. The class C mutant, in this case, the proteins gets accumulated in these vesicles which bud off from the ER and then stay there. But in the normal case scenario, they were supposed to go to the Golgi and get fused. But in case of sec C mutants, it is SECC. You got that? In this kind of mutant, they stay in these vesicles. Okay. The class D mutants, they basically get accumulated in the Golgi compartments. We can still see some here because obviously at any given point of time, all the secretory proteins are being synthesized in the ER and then being transported to the cis Golgi, but they are still here. Like some proteins will still be there because it is not 100% at one particular point. Okay, so that is why we can still see some here. Anyway, so majority of the proteins will remain stuck in the Golgi. Rather, they were supposed to form vesicles from the TGN and to be transported to the plasma membrane, but they don't. That was their normal scenario. Transport from Golgi to secretory vesicle, excuse me. Yes. So they were supposed to be taken to the secretory vesicles, but in this class D mutants, they stay back in this Golgi body. In the class E mutants, they get stuck in these secretory vesicles, but in the normal case scenario, they could have actually got fused, they could have gone to the cell surface and fused at the cell surface. But in this kind of mutants, at restrictive temperature, the proteins remain stuck in the secretory vesicles. So from this, from all these 5 mutants, and they are all in chronological order. So from here, the protein goes here. From here, the protein goes here. From here, the protein is supposed to go here, and then from here, go here, and stuff like that. So what happened? So we actually, through all these sec mutant experiments, we got to know what is the fate of secretory proteins in normal case scenario. They get transported into the ER, bud off from the ER, goes to the Golgi, goes to the secretory vesicles from Golgi, get fused with the cell surface. Okay, in normal case scenario. And in case of these defective mutants, the proteins will stuck at different phases. Now, the question is, if we have double mutant, it is quite possible that we have double mutant. In that case, what will happen? So let's say there is this class B mutation as well as class D mutation in one particular cell. So it's a double mutant. In this case, where will the proteins, secretory protein stay? Will it accumulate in the rough ER or will it accumulate to the Golgi? Remember, it's a stepwise process. A to B, B to C, C to D, D to E. If something happens at the B, it won't even go to D, right? So that means if it is a double mutant, let's say B or D, the first mutation point, here the proteins will get accumulated. If it is a B and E mutant, then obviously the proteins will remain accumulated in the ER. If it is A and C mutant, the proteins will remain in the cytosol. Okay. So study with sec mutants confirms secreted proteins are synthesized, processed, and move sequentially. Okay. Go to the next slide. Excuse me, totally forgot to switch on the mic. So we are talking about the cell-free assay, which demonstrates protein transport from one Golgi cisterna to another. So far we have discussed the protein movement from ER to Golgi to cell surface or from ER to cis Golgi. Now we are going to discuss what happens within the Golgi network itself. That means cis Golgi, medial Golgi, trans Golgi. So how in the first place scientists came up with this knowledge that there are actually three different kinds of compartments and how the compartments are identified based on the specific. So far we know that compartments can be identified based on the specific enzymes in those compartments. So if we change the inside nature of the compartment, we'll basically change the compartment itself. Okay. So in this case, we have used the VSVG protein. There is the wild type VSVG infected wild type cells. We said infected because this is a viral protein, but these proteins gets produced in the ER and goes to the cell surface because these are surface proteins. These are wild type cells, so it has to go through the normal cisternal maturation and eventually go through the secretory pathway to the cell surface. This one is VSVG mutant, infected mutant cells. In this particular mutant, this particular enzyme called N-acetyl glucosamine transferase 1 or NAG1 is missing. Okay. Now, why are we taking a cell, mutant cell, where this enzyme is absent? The reason is, remember in the previous experiment what we discussed, that from the ER to cis Golgi, 3 mannose residues gets deleted or reduced or cleaved. Now we see 5 mannose residues here and the NAG units. So now what happens when it goes to the medial Golgi, this particular N-acetyl glucosamine transferase 1, because it's transferase 1, so it transfers one NAG unit, that is N-acetyl glucosamine unit, to one of the mannose residues. Okay, so that particular NAG residue gets added to the one of the mannose residues. That is the change or the maturation of this oligosaccharide when the whole protein, oligosaccharide protein, goes to the medial Golgi. When it goes to the trans Golgi, further modification happens. We are not going to discuss about that, but further modification happens where several other units gets added along with uh, excuse me along with N-acetyl-neurominic acid. Also, we get galactose addition, these orange structures or circles. So if this step doesn't happen, this won't happen. So there is no question of having this maturation here. So these maturations are very specific to individual compartments based on the specific enzymes present in those compartments, right? So in this mutant cells, since it lacks this particular transferase 1 enzyme, so there is no addition of this unit, that is NAG unit here on the mannose residue. If this doesn't happen, the third step, that is the addition of further units, won't happen here. So it is also sequential, A to B to C. If something gets stuck at that, won't go to the C, right? And so far we know that enzymes are very specific to the compartments. Now let's do a fun experiment. Let's take isolate Golgi's from VSVG infected mutant cells. There is this green in structure mutant cells and non-infected wild type cells. This one. This doesn't have any any protein, VSVG protein oligosaccharide, just the Golgi compartment itself. Okay, Golgi compartment means all different sorts of Golgi compartments. So without the presence of any proteins. So, but these compartment specific enzymes, remember that. Okay, so this is supposed to have this transferase 1, N-acetyl glucosamine transferase 1 enzyme. So when we fuse these together, incubate them together, what happens? The vesicles bud off from this wild type cells, which contains the enzymes from this medial Golgi, and then those smaller vesicles with this N-acetyl glucosamine transferase 1 enzyme fuses with this Golgi, which lacked the N-acetyl glucosamine transferase 1. Remember the nature of the vesicle changes. So now it becomes wild type since we have now the presence of this N-acetyl glucosamine transferase 1 in this new cell. And now it should be able to add this particular unit, N-acetyl glucosamine, to G protein. And then it should be able to go to the next trans Golgi network to get processed. Okay. Excuse me. We discussed about G proteins, and I mean, we discussed that you all know G proteins, but just to be sure, G proteins, also known as guanine nucleotide binding proteins, are a kind of family of proteins that acts as molecular switches inside the cells. They can be turned on or off and are involved in transporting or transmitting signals from a variety of stimulus, outside stimulus, to a cell. Remember one particular set of proteins through this regulated secretion pathway. These are the kind of signal proteins that receives the outside signals, the G proteins, and then they affect the inside path. Okay, so excuse me. So here we can see that presence or absence of enzyme can make a huge difference. Also changes the nature of the Golgi compartments itself. So the conclusion is N-acetyl glucosamine transferase 1 moved by transferred vesicles from wild type to medial Golgi cisterna to the mutant cis Golgi during cisternal maturation process. Okay. Now the question is, what happens to modified proteins which goes to the trans Golgi in a wild type, or what happens to unmodified proteins rather? So let's say if a protein remains unmodified, like all the enzymes are there, but somehow some protein gets skipped, did not get processed here. What will happen? Whether they will go to the trans Golgi? No, they won't. What will happen to the fate of the protein? Remember the retrograde transport? They will go back to the cis Golgi and then again come to the medial Golgi, or rather becomes mature to become the medial Golgi. Excuse me. Now, how will you see the actual difference between the proteins? Obviously, because here we'll have extra units added, here we don't. Here we have a lot more units added, here we don't. So when we run it on the gel, based on the movement, based on the heaviness of the protein or the size of the protein and the oligosaccharide, obviously, we can see the difference. Okay, we'll stop here and go to the next section in next recording. Okay, bye. Hello students, welcome back to this section. Before going to the next section, so we stopped here pretty much. Here is my pointer. So yeah, so we stopped here. I remember in my second or third lecture, I guess, I used the other pointer which did not indicate anything. So you were not able to see the cursor or the pointer rather. I'm sorry about that, but I'll make sure that it doesn't happen again. So we stopped here and we saw that every compartment of Golgi network have their specific characteristic feature, that is the presence of specific proteins, compartment specific proteins, which gives the compartment the nature or the identity. Okay, and that those compartmental proteins gets transferred from one compartment to another compartment through vesicle bud off, which also carries those specific compartment specific proteins and gets fused to the next one, next compartment, and makes that compartment a new compartment rather, uh, because of the accumulation of more and more of that proteins. We have experiments to show that, but I don't think that we are going to talk about that in this portion. Anyway, going to the next section, this is vesicular traffic secretion and endocytosis. But before beginning, you might see that the previous section was a little longer, about 1 hour. So one might say that why 1 hour, why not smaller? Well, for several reasons. The first is this is a new opportunity. What is new opportunity? Well, this is an online class. We are basically bound by time in regular classes. 1 hour 15 minutes is not good enough to go over the syllabus and to explain each details, although I try to have as much detailed annotation as possible on the slide so that students do not need to go over any kind of materials, written materials. The pictures and the slides should be good enough, uh, but still, since we have got this opportunity that we can listen to the recording at any point of time of our choosing, so why not? I mean, I can explain it as detailed as possible, and so that you don't even need to go over the material from the book if you don't want to, because everything will be discussed during these. And it is the effort from my side that I actually have to talk about all these. But anyway, I enjoy this. As long as it helps students, I enjoy this. And I think so should you, because you are saving a lot of time, like driving down to the university, running around the classes, and then going back and then not having enough time to eat and stuff like that, you can just sit at home and then enjoy the lecture. If you really enjoy it, I mean, you'll love it, because I think that there are certain sections, or not even certain sections. I mean, for me, because I'm in love with molecular biology, it has been my life for a long, long time. So I mean, for me, pretty much every single topic, I'll say that it's pretty interesting. And you might get bored at the end saying that. You say that all the time. Well, because I actually feel that every single topic is so important and so logical, because everything flows with logic. If you see that anything is missing, the system will stop working. That itself is like such a precise, system. Anyway, so this particular section talks about the vesicular traffic secretion. So far, we have seen that how the vesicle bud off, and then how the proteins move from one compartment to another. So vesicles do form in the anterograde transport, which is only from the ER to cis Golgi. I said only from ER to cis Golgi. Why I said only? Because there is no other anterograde vesicle formation, because cis Golgi to TGN, it's actually maturation of the previous compartments. There is no vesicle formation. But in the retrograde transport, there are vesicle formation from TGI till the ER, because those vesicles have to carry those specific compartment specific enzymes to its below compartment. So now the thing is, obviously, the cell cannot confuse by having similar kind of vesicles, right? So vesicles, whether they are for anterograde transport or for the retrograde transport, or from one organelle to another organelle, like TGN, like ER to cis Golgi, or TGN to plasma membrane, or TGN to late endosome or lysosome, all these vesicles have to have some kind of identity, right? Some unique features. Well, those identities are actually being provided by specific coated vesicles or coat proteins. Okay, there are total of 3 types of coated vesicles. Okay, excuse me, COP2, COP1, and AP complex. We'll talk about that. Then there are involvement of these small GTPase proteins which direct the coat protein polymerization. So if anything happens with the small GTPase, those are specific. If anything happens with these GTPase, coat protein won't even form. So if the coat protein doesn't form, the vesicle won't form, and there is no question of trafficking the protein from one place to another. And then the coat protein forms, that means the polymerization of coat proteins around the vesicle happens. But at the same time, it also get released or depolymerized. That means that is called coat shading. Coat shading also happens because the function of coat protein formation is just to recruit necessary proteins inside the vesicle onto the membrane of the vesicle. Once it is done, once the function is achieved, goal is achieved, the coat proteins basically has to shed so that the vesicle can get fused to the next compartment. So that is the basis of this particular section. Let's go to this coat formation itself. Okay, or not even coat formation, rather the general structure of the vesicles and the coats. So this whole structure is a coated vesicle that is getting bud off from this donor membrane. Okay, so let's say this is a donor membrane, and this one is the target membrane. So we can see, so the donor membrane can be anything. So let's say the protein is moving from the ER to cis Golgi. So the donor membrane will be the ER membrane, and the target membrane will be the cis Golgi membrane. So let's say the vesicle is getting bud off from the donor membrane, that is ER membrane for the time being. So this particular membrane of the vesicle will actually be the membrane of this ER membrane, same membrane. Okay, but there are these small GTPases, blue in color. These are GTP binding proteins. These proteins gets recruited onto these budding of vesicles, These GTPases then further recruits these green proteins. Those are called coat proteins. These coat proteins are very specific depending on what kind of vesicles are getting formed, whether they are for the anterograde transport or for the retrograde transport or for the lysosomal or endosomal transport, what kind of transport. Based on that, particular coat proteins will get particular GTPase proteins will get recruited onto the membrane of the burning vesicle. Also, those particular GTPases will recruit particular coat proteins, and then those particular coat proteins will further recruit certain membrane proteins. For example, these V-snare or vesicle snare proteins. Okay, sometimes these membrane cargo proteins. Okay, and then sometime membrane cargo protein receptors. So membrane cargo proteins are the proteins which remains part of the membrane itself. Okay, those are recruited from this. These membrane cargo proteins earlier used to be part of these membrane, the donor membrane, which now becomes part of this vesicle membrane, which when after fusion to the target membrane will become part of the target membrane itself. Sometimes these membrane cargo proteins also becomes membrane cargo receptor proteins, and they actually bind the necessary soluble cargo proteins, which are these, the diamond shaped purple structure. These are the actual soluble cargo proteins, the most important point here, the most important part, which needs to be transported to the other compartment. Okay, so once this assembly happens and once the recruitment of these proteins happens, then there is an also this pinching off of this vesicle happens so that it becomes an independent circular vesicle. Then there is no need of the coat proteins anymore, because the purpose of the coat protein polymerization was to recruit these proteins. Once these proteins are recruited, then we can depolymerize or shed the coat. Okay, and once we shed the coat, we will expose these V-snare proteins which are embedded in the vesicle membrane. And only when the coat protein gets shedded or depolymerized, then only this independent vesicle can go near the target membrane, and then V snares will recognize similar target snare proteins, also known as T snare. They will actually they couple together, they basically coil together and form coiled coil alpha helix structure we'll discuss. And then this vesicle is pulled towards the target membrane. In this case, I was giving the example of cis Golgi. So this vesicle but off from the ER will get pulled towards the cis Golgi and will get fused. As it fuses, all these membrane cargo proteins will become part of the cis Golgi membrane protein, and these soluble cargo proteins will get released to the cis Golgi. Okay. So the vesicle buds are approximately 50 nanometer in diameter, and as I mentioned that recruitment of GTP binding G proteins to a region of donor membrane, the G protein, the characteristic feature of the G protein and the coat protein actually defines the nature of the vesicle. Okay, going to the next slide. As I was mentioning you that associated GTPase plays the most important role, and then based on the associate GTPase, it is going to recruit specific kinds of coat proteins. So for example, for the anterograde transport, that is from the ER to the cis Golgi, the vesicle coat type that gets formed is called COP2. Okay, so COP2 coat proteins rather, those defines the vesicles which are to be transported from ER to cis Golgi. And in those COP2 kind of coat proteins, the associated GTPase is called SAR1. This is a small GTPase. The SAR1 recruits the further coat proteins known as Sec2324, Sec1331, and then Sec12. Okay, Sec12 is already there anyway. But what about the retrograde transport? So if the vesicle has to burrow from the cis Golgi to ER or from the earlier Golgi compartments to the later Golgi compartments in the Golgi cisternae, what happens? The nature of the vesicle that forms is called COP1 coated vesicle. This COP1 coated vesicle has associated GTPase called ARF. These are all monomeric GTPases, that is single unit. So the ARF now actually recruits the cautamers containing 7 different COP units. We are not going to go into details of that. Okay, the main is this one. This recruits 7 different COP subunits. The third is clathrin and adapter proteins, also known as AP. Okay, so this is formed when the vesicle has to burrow from trans Golgi and have to go to endosome, or sometimes from plasma membrane to endosome, that is for the endocytic pathway. If you remember pathway 9 in the general secretory and endocytic pathway, sometimes Golgi to lysosome or melanosome or platelet vesicles. So different kinds of coat proteins gets recruited, but in all of these, the common factor is associated GTP is ARF. So in this case, clathrin plus AP1 complex trans Golgi to endosome, sometimes clathrin plus GGA. Now plasma membrane to endosome is clathrin plus AP2 complex. Well, how many of these you have to memorize? Good question. Pretty much everything. Why? Because we'll come across all these steps later on and you'll realize that this whole row is one particular diagram and you will understand the importance of all these molecules. So let's not decide on memorizing this one for the time being. Let's go to the next slide to see how actually the coat protein formation happens, especially in case of COP2 coat. Remember, COP2 coat is formed on top of the vesicles which are to be taken from ER to cis Golgi, that is for the anterograde transport, and the GTP is present in those COP2 coated vesicles is called SAR1, that is this one. Excuse me. So SAR1 is in GDP form. Okay, it's a GTPase protein. It is in GDP form when it is isolated and in the cytosol. There is a membrane-bound protein. There is ER integral membrane protein called Sec12 that is already there in the ER membrane. This recruits these SAR1 GDPs. And as soon as these SAR1 GDPs bind to the Sec12, Sec12 does a Jeff activity. Now you can say that, well, earlier we have seen in the nuclear transport, Jeff activity happens in the nucleoplasm and gap activity happens in the cytoplasm. Well, it is Jeff-like activity. Okay, Jeff-like activity means replacement of GDP with that of GTP. Okay, so GTP replaces the GDP bound to the SAR1, and now the SAR1 becomes GTP bound. As soon as it binds to the GTP, the conformational change happens, and now the SAR1 GTP gets released from this Sec12 and binds its N-terminus hydrophobic tail and insert it in the ER lumen. Okay, so now we have SAR1 GTP which is inserted in the ER lumen. So it is not going anywhere, it is stuck there. Now it starts the recruitment of the COP2 coat assembly. So in the process, the first set of proteins that gets recruited is called Sec2324 proteins. Okay, this is pretty interesting because these proteins are basically helpful in the recruitment of sorting signals. Those sorting signals recruits soluble proteins, and we'll discuss that in the later slide. So the Sec2324 gets recruited with the help of these SAR1 GTP version, and then a next set of code assembly happens that is called Sec1331. That is the second assembly of code proteins. That is not shown in this picture. I just mention it here. So after both the sets of assemblies happens around the vesicle, the vesicle now pinches off from this ER membrane and becomes independent vesicle with all the code protein assembly. Now what happens is that the Sec23 from the Sec23-24 complex, this initiates a gap activity. What is gap activity? Gap activity is basically the hydrolysis of bound GTP to the SAR1. So we know that SAR1 now is GTP version. So the Sec23, which is bound by recruited by SAR1, actually helps in the hydrolysis of the bound GTP. Once it basically hydrolyze the GTP, it provides enough energy to depolymerize all these coat proteins around the vesicle. Now this becomes uncoated vesicle after the coat disassembly. And remember, the whole process of coat protein assembly was to recruit all these proteins, right? All these cargo proteins and the soluble proteins. That has been achieved. So now there is no need of these coat proteins, so they get shed from the vesicle. Now the vesicle has exposed V snares or vesicle snares. Okay, excuse me. Now what happens to cells expressing mutant SAR1 or ARF in that sense that is unable to hydrolyze GTP code? So for example, if these SAR1s, if the Sec23 is unable to hydrolyze this GTP here, what is going to happen? Think. Since the hydrolysis provides energy to release all these coat proteins, if the hydrolysis doesn't happen, this vesicle won't have energy, so the unpolymerization or depolymerization or the coat shading won't happen. If the coat shading won't happen, these V sneers won't get exposed. If the V sneers don't get exposed, the vesicle will not be able to fuse with the target membrane. So although the coat formation happened, the coated vesicle formation happened, all these cargo proteins that is to be carried to the next compartment, all those are recruited, but unless and until the coat disassembly happens, it is basically of no use. Okay. So why is Sec23, Sec24 important? Obviously, if you see the crystal structure, this is the crystal structure, three-dimensional structure of the ternary complex comprising of the COP2, coat protein Sec23, 24, and SAR1 GTP. So this red protein here is the SAR1 GTP. The small GTP is this is the cytosolic side. And this is the ER membrane in this case. So you see that the SAR1 obviously has a tail here, which is not shown. The SAR1 recruits the Sec23 and 24 in the first step of coat polymerization. The Sec23 is nearby SAR1. That is why Sec23 is responsible for the hydrolysis of GTP bound to the SAR1. There is also associated Sec24. Why is that important? Well, the Sec24 interaction with the cytosolic domain, that is this cytosolic domain of diacidic tripeptide diacidic sorting signal. This is a sorting signal. This recruits the cargo proteins in the vesicle membrane. So this sorting signal is being recruited by Sec24, and this particular sorting signal, this is the hydrophobic portion embedded in the membrane. This sorting signal recruits specific cargo proteins which are to be transported to the next compartment, which in this case is the cis Golgi. Okay, so this is the transmembrane segment of cargo protein. Now, if this sorting signal is not being properly recruited by the Sec24, then we cannot recruit a specific cargo protein itself. That is why binding of Sec24, right, recruitment of Sec24 by SAR1 is extremely important. Sec24 recruiting the tripeptide diacidic sorting signal is extremely important so that this sorting signal can actually bind the specific cargo protein to be transported. Okay, and remember, all these cargo proteins are secretory proteins. So we know about one particular disease called cystic fibrosis, right? Cystic fibrosis, the lungs get congested and people have breathing issues, and lungs get filled with cough and mucus and all those stuff. We'll discuss in a later slide maybe. Just to give you some idea, excuse me, in the cells in lungs and other organs which misses the CFTR plasma protein. So basically in this cargo protein, in the case of cystic fibrosis, this cargo protein is called CFTR protein, and that is a plasma protein, so plasma membrane protein. So this cargo protein gets recruited by the sorting signal, and then sorting signal, so this vesicle gets fused to the cis Golgi and then further gets matured as it goes through the trans Golgi network, and further it goes to the plasma membrane of the cell in case of lungs, alveolar cells. So CFTR contains a diacidic sorting signal which delivers the CFTR protein to the plasma membrane by constitutive secretory pathway. But there is a mutation called delta F508. Okay. So at 508th position, there is a deletion mutation, which is the major cause of cystic fibrosis. So misfolded cytosolic domain. So in this case, the misfolded cytosolic domain prevents the CFTR recruitment, okay, into the ER COP2 coated vesicle because the sorting signal is misfolded in case of delta F508 mutation. In that sense, the CFTR protein cannot be taken to the plasma membrane, and CFTR proteins remain inside the ER lumen and eventually degraded. Okay, so that is the basis of the cystic fibrosis. Anyway, going to the next slide. So we have seen that the coat proteins are important. Coat proteins are formed based on the nature of small GTPases. Those recruit specific coat proteins. The vesicle formation happens. Depolymerization of the coat happens. Now the exposed V smears are present on the depolymerized vesicles, decoated vesicles. Okay, how the vesicles are going to get fused to the target membrane? So this is a very generic example which shows the docking and fusion of transport vesicles with their target membrane by the presence of RAB GTPase. This is another small GTPase like RAS. It is actually a small GTPase from the RAS superfamily itself. So these proteins are present in the cytosol. They get recruited, excuse me. These basically they are tethered via lipid anchor to the secretory vesicle. So these are the secretory vesicles. This is the RAB protein, RAB GTP version. These are tethered to the membrane by a lipid anchor. Okay. Now what happens? We have these V-amp. These are the V-snear proteins. Specifically, these are called V-snears or V-amps. And now we have the target membrane which has got a RAB effector molecule. The RAB effector molecule basically bind these RAB GTP, recognize and bind RAB GTP. And on the target membrane, we have T smears. In this particular example, they are called SNAP25, two of them, and another one called syntaxin. All of these are T snares or target snare protein. So once this RAB effector binds the RAB GTP, they basically RAB GTP is bound to this whole transport vesicle. They come close together. As they come close together, these V snares on the target vesicle, I mean the vesicle itself, secretory vesicle, they coils with the T snares on the target membrane. This is called snare complex. Now, the snare complex, as it happens, as it forms, they form four coiled-coil interaction. And then this vesicle slowly fuse with the target membrane. Now, exact process is not explained because that will be too much detail. Now, there are certain proteins called NSF and alpha SNAP. NSF is N-ethyl maleimide sensitive factor, and alpha SNAP is soluble NSF attachment protein. These particularly, so these basically binds to these coiled coil complex, and this, remember this RAB GTP was bound to the RAB effector. The hydrolysis of GTP happens, and the hydrolysis produces enough energy to release this RAB protein from this structure, bound structure, and this RAB gets recycled back for another cycle of assembly or fusion. Okay, so RAB GTP hydrolyzed by RAB GDP, hydrolyzed to RAB GDP, which dissociates the RAB from the RAB effector. That particular step is not shown in this case. So going to the next slide, this is just a structure which shows how they are arranged. So there is one alpha helix, that is VM in this case. Excuse me. And we have one alpha helix called syntaxin that is associated with the membrane itself. Okay, the target membrane itself. This is the vesicle membrane. This is the target membrane, and there are two T snares called SNAP25s, two alpha helices. These SNAP25s are anchored to plasma membrane by hydrophobic lipid anchor. Okay, so supposedly, if in this case we are not talking about, we are talking about the plasma membrane. So basically, the vesicles which bud off from the TGN and goes to the plasma membrane, we are talking about this particular. So in case of plasma membrane, we have these SNAP25s anchored to plasma membrane by hydrophobic lipid anchors. Okay, so four long alpha helices, two from the SNAP25 and one each from syntaxin and VAMP, form numerous non-covalent interactions here to form four coiled coil helix, and these holds together to bring this coated vesicle or uncoated vesicle or depolymerized. I mean, there is no coat basically. So those vesicles they bring to the target membrane to get it fused, and in the meantime, a RAB basically hydrolyze the GTP to get energy to get released to get recycled back. So that is the whole process of how the actual coat assembly happens, the vesicle formation happens, and the fusion happens. So we'll stop here and we'll go to the next section in a bit. Okay, all the best. Hello students. Uh, let's come to this third section of the fourth lecture. This is the last section, I promise. So we stopped here and we saw that how the vesicle gets fused and how the coiled-coil helix forms with the target membrane, the V snares and the T snares. So coming to the next section, we'll talk about the stages of secretory pathway and the COP2 and COP1 coated vesicles, and basically the receptors, the sorting signals. Okay. So anterograde transport, we all know by now that it is the transport of proteins through a COP2 coated vesicles from raphia to the cis Golgi, and retrograde is through the COP1 coated vesicles. The vesicles come back to the previous compartment which supports the Golgi cisternal maturation, obviously. Also, if some missorted or misfolded proteins reaches to the cis Golgi by mistake, they are taken back to the ER through these COP1 coded vesicles as well. Okay. So now coming to the vesicle mediated protein trafficking between the ER and cis Golgi. So the general process that we have discussed so far is through the COP2 vesicle. These are the membrane proteins like membrane receptors and the membrane cargo proteins. The membrane receptor proteins recruits the soluble cargo proteins, and the membrane cargo proteins are basically part of the membrane, the proteins that are present in the membrane. So these are part of the already existing ER membrane. And then when what is the GTPase that gets recruited in case of COP2? SAR1. Okay, so SAR1 basically recruits the coat proteins, in this case, the 2324 and 1331 in 2 stages, and also helps in the recruitment of specific soluble proteins or the cargo proteins, and then it buds off. Basically, it gets pinched here. We'll discuss that later. It gets bud off and then forms a complete vesicle. The COP2 coat gets depolymerized to expose these V sneers. The V sneers combines with T sneers, and then basically this vesicle fuses with the target membrane, in this case, cis Golgi. Now, as the vesicle becomes the part of the cis Golgi itself, vesicle means the vesicle membrane becomes the part of cis Golgi network, we are transferring all these membrane proteins to the cis Golgi now. So we are now in shortage of those membrane proteins in this membrane. Also, uh, the area of the membrane, basically area of the rough ER membrane is getting reduced because we are basically budding off portions of the membrane which gets fused here. So we need to bring back those membrane proteins as well as the membrane portions itself. How does it that is done is, again, vesicle gets formed here, gets bud off from the cis Golgi network. Through retrograde transport, the vesicles transfer the proteins to the previous compartment, that is the ER. And this time, there is a specific kind of coat protein formation happens. These vesicles are called COP1 coated vesicles. This is COP2 vesicles, coated vesicles for the anterograde transport. COP1 coated vesicles are for the retrograde transport. So here, all these membrane proteins, like most of it, they are already part of the membrane, and they also recruit all those missorted or misfolded proteins that accidentally skipped here from the ER. They collect all those, and then these vesicles gets bud off, then gets obviously depolymerized, the coat protein gets recycled, and then this vesicle gets fused with the T sneer and V sneers. So now the T sneers will be on the target membrane, that is the ER membrane. And as the membrane proteins again becomes part of the ER membrane, that is how. So the importance of retrograde transport is obviously for the retrieval of V sneer proteins, also for the retrieval of the missorted ER resident proteins, as well as retrieval of membrane bilayer. So that is also very important. And this is for the retrograde transport between the cis Golgi and the ER. For the COP1 coated vesicles, in case of retrograde transport, in case of cisternal maturation, those brings back the specific compartment specific enzymes, which we are not discussing here. Okay. Now going to the next slide, we discussed, if you remember, we discussed about diacidic sorting signal in case of the COP2 coated vesicle. So those diacidic sorting signals were being recruited by the SEC24, which is being recruited by the SAR1 during the formation of two coated vesicle. So this is the example of diacidic sorting signals. So cargo membrane proteins in the ER. Okay. The membrane proteins is COP2 coated membrane COP2 coated vesicles, and it is being recruited by the SEC20 subunit. Now, this whole chart, do we have to remember all of these? Not really. We need to know few specific ones, especially the KDEL, because we have a whole description of KDEL. M6P is extremely important. We'll see later on why. We have already discussed about diacidic sorting signals. Diarginine sorting signals also becomes important. We'll see later on. And also this MPXY signal, we have a complete section on this one, the LDL receptor. So I haven't highlighted the NPXY signals and the diacidic sorting signals. So we are left with these two and probably this one. So anyway, kind of all of them are important to me. Going to the next slide, this one talks about the KDEL signal. Sorry, I totally missed this pointer. I just remembered. So we will be discussing about this KDEL signal. Okay. So if we go to the next slide, this KDEL receptor is this. So let's discuss what is important about KDEL. First of all, KDEL is associated with COP1 vesicles. From that, based on the informations we have, COP1 vesicles are formed during retrograde transport. That is, when the vesicles bud off from the cis Golgi to the ER. The KDEL receptors in cis Golgi membrane, basically they are present in the KDEL receptors are present in the cis Golgi membrane itself. Now, KDEL sequence is basically the names of the short forms of the amino acid itself. So lysine, aspartic acid, glutamic acid, and leucine, KDEL. Okay. So KDEL is a sequence. It's a small peptide. It's a 4 amino acid peptide. It's a small sequence which is attached at the C terminus of the proteins which are present in endoplasmic reticulum. Okay. KDEL receptor is present in cis Golgi membrane. Now KDEL receptor binds to the KDEL sequence which are associated with the proteins. That means the KDEL receptor basically recruits all the proteins which have KDEL sequence, and through the retrograde transport brings those proteins with the KDEL sequences at the C terminus back to the ER. Now, having this information, this is how it looks like as a pictorial depiction. So these red color signals, these are basically KDEL peptide. These are the sequences of the C terminus, that is C terminus of ER targeting sequence. Okay, these are associated with the protein, right? Or the peptide. These peptides are basically luminal proteins. Okay. So luminal proteins, ER luminal proteins means they are supposed to stay inside the ER all the time. They are not supposed to get into secretory mode and get outside. They're supposed to stay inside the ER. Somehow, if the proteins gets into these budding vesicles, in that case, the budding vesicles will take them eventually to the next compartment, that is the cis Golgi compartment. Now we have these missorted ER resident proteins. The ER resident proteins are supposed to stay inside the ER with the KDEL peptide. At the same time, we are seeing that we have these KDEL receptors. These are KDEL receptors on the cis Golgi membrane. Now, these KDEL receptors recognize and binds these KDEL peptide and in turn the whole missorted ER resident protein itself. And through the formation of COP1 coated vesicles, they through the retrograde transport come and get fused with the ER membrane. Now you can say if the KDL receptor is a membrane protein, this is the KDL receptor. If it is a membrane protein, why is it functioning only in the, why is it recruiting the peptide with KDL sequence only in the cis Golgi and not in the ER? Well, because of this difference. There is lower pH and higher hydrogen ion concentration in the cis Golgi, which helps in the peptide binding by this KDL receptor to this KDL peptide. In case of ER, we have higher pH. So higher pH causes peptide to release from the KDL receptor. So when it comes back and gets fused with the ER, the higher pH in the ER basically helps in the release of the resident proteins from the KDL receptors. The KDL receptors, remember, is still bound to the membrane because those are membrane proteins. And eventually, those KDL receptors are brought back to the cis Golgi compartment through the anterograde transport, that is COP2 coated vesicles, because KDL receptors are membrane proteins. So this here, due to the high pH, the peptides with KDL sequence gets released in the cis Golgi compartment. The peptide with KDL sequence gets recruited by the KDL receptor due to the low pH. Okay. These KDL receptors have KKXX sorting signals at C terminus. Okay, that is on the cytosolic side, binding to COP1 alpha and beta subunits. 2 for COP1 coat for retrograde transport. So these membrane proteins have this KKXX sorting signals at the C terminus and binds to COP1 and alpha and beta subunits. So COP1 alpha beta subunits basically have these KKXX sorting signals which recruits these KDL receptors and the KDL sequence attached proteins itself. So what happens in case of temperature sensitive yeast sec mutants lacking COP1 and alpha and beta subunit? It's a good question. So in this case, if they lack COP alpha and beta subunits, obviously, although the KDL receptor can bind these missorted proteins with the KDL sequence, the COP1 vesicle, the coats will not form. So COP1 coated vesicle will not form. So the missorted proteins won't be able to return back to the ER compartment. They will stay in the cis Golgi compartment itself. Another sorting signal is diarginine sequence can reside in any segment of membrane protein on the cytosolic side. So we have discussed about that in the previous SAR1 recruitment with SEC23. Yeah. So, excuse me. Everything here is so logical. Now, let's say that since we know that any peptide with KDL sequence will become the resident ER resident protein. Now let's say that we need to do an experiment where we need to keep a protein which is a secretory protein. We need to keep that protein inside the ER all the time to make sure that it doesn't escape the ER or doesn't get secreted outside the cell. What can we do? we can just add KDEL sequence at the end, at the C terminus end of that specific protein that we need to localize in the ER compartment. That's it. So it is pretty intelligent system in itself, that if something gets escaped, it will come back. Also here, although the receptors are present, it won't bind the peptides because of higher pH. Here it binds the peptides because it has lower pH. That's awesome. Anyway, going to this, the next segment, this is just an example to show you what happens in the cystic fibrosis. We discussed that before. And what happens to the cystic fibrosis is that imbalance in the chloride ion transport in the epithelial cells of lungs causes the fluid buildup, and it causes difficulty in breathing. So this is the normal CFTR channel. It helps in the movement of chloride ions from inside the cell to the outside. But in case if this channel is absent or mutated, in this case this is a mutant CFTR channel, it cannot release the chloride ions from inside to outside of the cell. And that causes the sticky mucus to build up on the outside of the cell. So it will eventually impact the gaseous exchange when we breathe in, and the cells won't get enough oxygen. The blood will not get enough oxygen and will cause breathlessness. Okay. So that is cystic fibrosis phenotype. Now, two things can happen. Either something happens that this channel gets mutated, which is this particular case. The other thing can happen is that there is certain specific mutation which doesn't let this channel even come to the plasma membrane. In that case, there is no question of chloride ion transport. So this is a normal cell where we have this Golgi body, and from here, from ER to Golgi, and then eventually the CFTR channel gets secreted to the plasma membrane, and which helps the secretion of chloride ions from inside to outside. This is the normal functioning of plasma membrane in the alveolar cells. Now there are two situations. In this case, we have delta 508 mutation. That means in the sorting signal itself. Okay. Sorting signal where? Sorting signal in case of COP2 coat formation. So the COP2 coat have some mutant sorting signal. That mutation is basically phenylalanine deletion at 508th position. In that case, it cannot recruit the CFTR protein. Although remember, the CFTR protein gets properly folded, and it's properly folded version in the ER, but it doesn't even get recruited into the COP2 vesicle. So it cannot even go to the cis Golgi and further to the plasma membrane itself. So although it's completely not functional, it's in a way completely functional and properly folded, but it cannot reach to the plasma membrane. So in that case, no chloride transport. That means we have this kind of situation. In this case, we have the chloride, I mean the CFTR channel here, but it is mutated. So again, no chloride transport. So nothing happens. So a CFTR, folded CFTR with this mutation, delta F508, will function properly as a normal chloride channel, but will never reach the surface membrane, which means that the diseased state is caused by the absence of chloride channel and not by the presence of the defective ones. Okay, so that is it. And two major models for intra-Golgi trafficking will end here. But we have been talking about this cisternal maturation for long. So people came up with theories. Some said that in one model, that is vesicular transport model, people said that, well, the vesicles gets brought off from the ER and becomes cis Golgi. Then again vesicles brought off from the cis to form these medial Golgi. If you see these cargo proteins, so vesicles basically takes these, recruits these cargo proteins, and the cargo proteins gets transported to these individual compartments. Okay, these individual compartments existed all along, and eventually the cargo proteins get secreted. These dots are cargo proteins. So Golgi proteins remains in the cisternae, that is these individual transcisternae, medial cisternae, and cis cisternae. Those are individual compartments, and the cargo proteins rather moves to or taken to the another compartment by vesicle budding. The other theory said, cisternal maturation process model, that these budding of vesicles from the ER forms the cis Golgi cisternae, and that's it. After that, there is no vesicle budding from the cis Golgi, which goes to fuse the cargo proteins here. Rather, what happens, excuse me, these trans Golgi network, these releases these clathrin coated vesicles back to this, which gets fused back to the trans Golgi network or trans Golgi, and takes all the enzymes. And then from here, through the COP1 coated vesicles, the enzymes are taken here. And from here, the enzymes are taken here. As they are transferring the enzymes, they are basically changing the nature of the compartment from cis to medial, medial to trans, and trans to trans Golgi. Now, if this eventually becomes trans Golgi, what will happen to the cis and medial? Remember, this particular layer formation is happening constantly. So by the time this one becomes the trans Golgi, there are two more layers that gets formed, so that it can transfer the materials here and then here. So it's a continuous process. So each layer gets formed and each layer gets the enzymes from the previous compartments. So that is called cisternal maturation model. So in this case, the cisternae themselves functions as the cargo carriers. So cisternae themselves as forms the cargo carriers. The nature of cisternae changes due to Golgi protein transfer in retrograde fashion. So eventually, people accepted this particular model, that is cisternal maturation model. And we have one particular paper as a reference. If someone wants to go over, then they can go over this particular paper and see the difference in these pictures. So I think I'll stop here for the lecture 4 and go over the material as many times as possible. We have the entire week, like today, Thursday, Friday, Saturday, Sunday, and then we'll come up with the next lecture on Monday. So all the best, stay safe and stay at home, and we will see you next week. Bye-bye.